Now we have an overview of the process by which all these different types of formed elements are made. Let's take a look at each of these formed elements a little more up close and see what they're all about. And we'll start with red blood cells. So just remind you again, the main or principal job of red blood cells is to transport oxygen out to the tissues, and it does this by filling itself full of hemoglobin molecules inside the red blood cell. Now, obviously, for oxygen to get into that red blood cell where the hemoglobin is located, it has to diffuse through the plasma membrane. And this leads me to the first characteristic of erythrocytes that makes them unusual among cells, and that is the fact that they have what's called a biconcave shape to them. And this biconcave shape, remember, concave means dipping, having a depression or dipping into. So you can see here that if we look at the cross section of this red blood cell, it does indeed have a biconcave shape. That produces a larger surface area. In other words, more plasma membrane is present in the cell than you would normally see for a cell with that volume of cytoplasm. And the result is that it means there's more plasma membrane through which oxygen can readily or rapidly diffuse into the cell and then attach to hemoglobin. So that's a very unique quality of erythrocytes. Another unique quality of your erythrocytes, remember, is that they are without a nucleus or anucleated. Remember, we ejected that nucleus during development in the bone marrow. If we look inside the cytoplasm of the erythrocyte, we can see that it's made up of, obviously, water, as all cytoplasm is, but it also has a lot of, on the surface of plasma membrane, glycoproteins and glycolipids that together contribute to the blood type. So on the surface of the cell, we're going to see later, are some little glycoproteins that help determine the type, whether you have type O, type A, type B blood, whether your blood is positive or negative. And whether you realize this or not, there's about 23 other different types of blood typing systems aside from the ABO and positive negative typing systems that also relate to the presence of these glycoproteins and glycolipids on the surface of the plasma membrane. Erythrocytes are highly flexible and bendable. This is a really critical feature of them because when they start passing through capillaries, and by the way, capillaries are the smallest among the blood vessels, the diameter of a capillary is so small that it is smaller than an individual red blood cell. So here's, like, for example, the diameter of a capillary. In order for that blood cell to get through that capillary, it's going to have to bend itself or fold itself over to move through. As erythrocytes age, as they get up closer to that 120 days old mark, they begin to lose flexibility. And this is usually a precursor to them being filtered out in the spleen and removed from circulation. Again, remember, there are pretty much no more functional organelles present in an erythrocyte by the time it is done developing. At the end of development, when it's mature, all the endoplasmic reticulum has broken down, the nucleus has been ejected. It pretty well is absent of most organelles. And so because of that, because there's no mitochondria present, for example, there's no way for erythrocytes to use aerobic respiration in order to make ATP. So they are the only cells in the body that exclusively utilize anaerobic respiration as the means by which they manufacture ATP. Actually, this makes good sense. If this is going to be the cell that transports oxygen out to tissues in the body, the last thing you want it to do is be consuming that oxygen on its way out to the tissues. So you don't want it using oxygen. You want it to undergo anaerobic respiration. Okay. The cytoplasm, as I said before, is made up principally of water, but it also has a lot of hemoglobin molecules in it. The hemoglobin molecules are those molecules that transport oxygen. In a couple slides, I'll show you more up close what the structure is of that hemoglobin. On average, there's about 280 million molecules of hemoglobin in each and every red blood cell. There's also another component of the cytoplasm that turns out to be really important, and that's this enzyme here called carbonic anhydrase. Carbonic anhydrase helps to produce something called carbonic acid, and that becomes critical when we start transporting carbon dioxide inside of a red blood cell. I'll show you more about that later on. The importance of iron in red blood cells cannot be overstated. Iron is critical because it is at the center of every hemoglobin subpart, and it is the iron molecule to which oxygen atoms bind so that they can be transported inside a red blood cell. So if you don't have enough iron in your diet, you are not going to be able to manufacture a sufficient number of functional hemoglobin molecules, and you can develop a condition called anemia. So I want to look at how it is we get iron absorbed into our body so that we can utilize it in the manufacture of red blood cells. When you consume iron-rich foods such as red meat or spinach, for example, that iron enters into your stomach here. Here's your stomach. And it can enter in two forms, either the Fe2 plus form or the Fe3 plus form. You'll remember this from inorganic chemistry. It turns out, though, that the small intestine, where we actually absorb the iron, you don't absorb it in your stomach, you absorb it in your small intestine. The small intestine can only absorb iron atoms if they are in the 2 plus state. All the iron atoms you consume that are in the 3 plus state have to first be converted to the 2 plus state so they can be absorbed. The stomach helps in the process of conversion of the 3 plus ions to 2 plus ions. You'll be happy to know I'm not going to ask you to know the process by which that occurs. You just need to know that the stomach is responsible for doing that. Once we get those Fe2 plus atoms down into the small intestine, then they can be absorbed into the blood plasma. And when they are absorbed into the blood plasma, they need a little help. And the help that they have is through a process of combining with something called apoferritin. Some books call it transferrin, but it's the same thing. This is a type of albumin. It's one of the types of plasma proteins. It combines with the iron atoms to help transport it in the blood, and it eventually moves it to the liver. And once it's in the liver, it's stored there until such time as it's needed in the bone marrow to make the hemoglobin molecules. So what exactly is that hemoglobin? Well, hemoglobin is a protein. And again, we said there's about 280 million molecules of this protein of hemoglobin in each and every single erythrocyte. And you have somewhere between, if you're a male, you have somewhere between 4.5 and 6.2 million erythrocytes per cubic millimeter of blood. So you can imagine the gazillions, if that's a correct word, of hemoglobin molecules present in your blood total. The hemoglobin molecule is a combination of 4 separate protein chains or globin chains. Okay, this makes a protein chain. Since it's made up of separate polypeptide chains, this means hemoglobin is an example of a protein that has quaternary structure, meaning that 4th level of structure. Remember, first primary structure is the amino acid sequence. Secondary structure are the alpha helices and beta pleats. Tertiary structure is how those helices and pleats fold in relationship to each other. And then if I get 2 or more separate polypeptide chains combined together to make a functional protein, that's when I have quaternary structure. Hemoglobin is made up of 2 alpha chains and 2 beta chains. So if we look down here, there are 2 alpha globin molecules and 2 beta globin molecules. These are very similar chains, the alphas and the betas. They just vary just slightly in their primary sequence. At the center of each of these globin molecules, here I'm circling it, it's this blue disc here. This blue disc represents what's called the heme group. And the heme group is a organic molecule, meaning it has lots of carbons present, but at the center of it, there's that iron atom. And it's that iron atom to which oxygen atoms attach. So now this heme group, each one of these hemes, here's one heme, here's another, here's a third, here's the 4th heme. Each one of those hemes has the ability to carry 1 molecule of oxygen. Remember, oxygen is a diatomic molecule. That means that there are 2 atoms of oxygen in each molecule, O2. Oxygen does not appear or occur by itself. So if I have 1 O2 molecule attached at each of these positions, that means that a single molecule of hemoglobin has the ability to carry a maximum of 8 oxygen, or the equivalent of 4 oxygen molecules. Okay, 8 oxygen atoms or 4 oxygen molecules. There's a interesting variant of adult hemoglobin. Okay, adult hemoglobin is that hemoglobin that has a beta and alpha configuration, 2 alpha and 2 beta globin chains. Fetal hemoglobin, on the other hand, has a slightly different configuration. In fetal hemoglobin, I still have 2 alpha chains, but instead of having 2 beta chains, I have 2 what are called gamma chains. Those gamma chains have a greater affinity for oxygen. Now, why would a fetus have a different type of hemoglobin? Well, think about it. Oxygen is delivered to that fetus via the placenta. So the mother inhales oxygen. That oxygen is attached to her hemoglobin molecules. The hemoglobin molecules travel to her placenta, and there they come up next to, they don't physically contact, but they come next to fetal hemoglobin molecules. Well, if the fetal hemoglobin were made of alpha and beta chains, same as mom's hemoglobin, they would not be able to have any stronger affinity for grabbing onto that oxygen, and the oxygen would thus remain on the mom's hemoglobin. By having fetal hemoglobin with these gamma chains that have a stronger affinity for oxygen, what this means is that the baby or the fetus's blood cells with its fetal hemoglobin will be able to pull the oxygen molecules off of mom's hemoglobin atoms and attach them to the fetal hemoglobin. So what do we do when a red blood cell is reaching the end of its lifespan, when it's getting out to that 4 months mark or about 120 days? Well, we rely principally on the spleen to clean out those red blood cells. And this is a multi-step process. The spleen, you'll remember, is made up of reticular connective tissue. And that reticular connective tissue looks like a meshwork of collagen, elastin, reticular fibers. And the red blood cells have to squeeze through that connective tissue matrix. And if they have begun to lose flexibility, as red blood cells do as they're reaching old age, they won't be able to squeeze through that mesh-like network of fibrous proteins, and as a result they get trapped in the spleen, and at that point macrophages, which is a type of white blood cell in the spleen, will come by and begin to degrade those red blood cells and to recycle the pieces and parts of those red blood cells so that those pieces and parts can be used to make new erythrocytes. So here's my macrophage, and here it is consuming a dead and dying red blood cell. And in this multi-step process, the first thing that happens inside that macrophage is when it opens up that red blood cell, it exposes the hemoglobin. And remember, hemoglobin is made of 2 parts. It's made of a heme group and the globins, those alpha and beta chains. So the first order of business is split apart hemoglobin into the heme group and the globin group. So here's my heme group, here's my globin group. The globins, remember, are just chains of amino acids, and we can break those apart into individual amino acids, and then those can be put out into the blood and utilized by any cell in the body that needs amino acids to make a new protein. The breakdown of the heme group is a bit more complex because that heme group contains an iron atom and it has all of these carbons and hydrogens associated with that iron atom. So the process of breaking that down involves the use of several enzymes. One of those enzymes will strip off the iron atom, attach it to that transferrin or that apoferritin, which is that transport atom in the blood, or transport molecule in the blood, and then we can shuttle that iron back to the liver for storage or directly to the bone marrow to make a new red blood cell, whichever is needed. The other portion of the heme group, once I've taken off the iron portion, I now have a green pigmented molecule called biliverdin. And that gets further converted with some help of some other enzymes into something called bilirubin. The biliverdin is green in color, the bilirubin is yellow in color. And that bilirubin eventually gets shuttled to the liver, and it gets conjugated or put together with some other molecules to form something called bile, which is then released into the intestines. It may eventually get altered through the process of digestion, and then it gets reabsorbed into the blood plasma. And eventually, long story short, if everything's going correctly, the kidneys will filter out that bilirubin, that yellowish colored pigment, and send it out in the forming urine, which is why urine has a yellowish hue to it. If something happens and a person's kidneys aren't functioning correctly, or if the liver isn't functioning correctly, people can get a buildup of that bilirubin, that yellowish pigment, in their blood, and it will start to deposit in their skin. At that point, they can get a condition that you probably know of, and that condition is called jaundice. That yellowish appearance to the skin. Let's talk very briefly about a few problems that can arise if I'm not making red blood cells appropriately, or if I'm making too many of them. The first condition we'll talk about is polycythemia. This means too many red blood cells are present. And the underlying cause of polycythemia can either be what we call a primary cause or a secondary cause. Primary causes mean that there's something actually wrong with the stem cells that make the red blood cells. They are dividing out of control, for example, and this happens very commonly with cancers of red blood cells, or what's called, it's a type of you wouldn't call it leukemia because it's with red blood cells. It's just a red blood cell cancer. How's that? It can be a cancer of the bone marrow itself, or just of the red blood cells. So secondary polycythemia is far, far more common. If something other than the stem cells themselves are causing the problem, then it's a secondary polycythemia. And you can get excess accumulation of red blood cells or secondary polycythemia for a number of different reasons. If you become dehydrated, then your hematocrit is going to increase, right? You've lost fluid, but you still have the same number of red blood cells, so technically you have polycythemia. You have too many red blood cells for the volume of blood that you have. People who smoke will develop polycythemia because literally there is the smoke inhalation is causing the inhalation of carbon monoxide. Carbon monoxide is present in tobacco smoke. Carbon monoxide outcompetes oxygen for binding to hemoglobin, and so to try to correct the problem, the body will manufacture more erythrocytes in order to grab onto oxygen atoms. Air pollution can also cause polycythemia. People who have a respiratory condition called emphysema can also get polycythemia. We'll learn more about emphysema when we get to the respiratory system. Living at high altitude can do it. We here in Flagstaff live at about 7,000 feet. I can tell you right now that the total number of red blood cells you have living in Flagstaff is greater than it would be if you lived at sea level, say in San Francisco. Again, that's because there's less oxygen at high altitude, so you need to have more red blood cells present to grab the oxygen that is available. Strenuous exercise can also encourage polycythemia. The idea here being that if your skeletal muscles are being put under a lot of stress, they need more oxygen. So we're going to make more red blood cells to deliver more oxygen to those muscle tissues. Okay. Other things can also cause secondary polycythemia, things like heart failure, I'm getting more into pathology here. If you want to know more about that, take pathology, Bio 320. Okay, so what kind of problems can arise as a result of having polycythemia? Well, obviously, you're going to have a greater viscosity of the blood because you've got more red blood cells present. And that can be a problem because the more viscous blood is, the more likely it is to slow down. The slower blood goes, the more likely you are to develop a thrombus or a blood clot. And that can also, that thrombus can later detach and move to another location in the body and become trapped, which is called embolism. That can lead to things like stroke. It can lead to things like myocardial infarctions or heart attacks. With polycythemia, usually your blood volume increases. And if you have higher blood volume, you're going to have higher blood pressure. Now, polycythemia can be artificially induced, and you might think, well, why in the world would anybody want to do that? Because there's all of these potential problems that can arise. But there can be benefits to polycythemia. really high caliber athletes, for example, cyclists, bicyclists, have been known to undergo a process called blood doping, where they either inject themselves with erythropoietin, that's the hormone that induces the manufacture of new red blood cells, or they, in the days leading up to a major race, will have their blood drawn. They'll have the plasma removed out of their blood so that all that's left is the red blood cells, and they'll have it stored. And then right before a major race, they'll have those red blood cells injected back into their blood. The idea being the more red blood cells I have, the more oxygen I can deliver to my muscle tissue, the better I'll perform. The downside, of course, is that, again, we're increasing viscosity. And there have been cases where bicyclists who have undergone blood doping have ended up dying because their blood became so viscous their heart could not circulate the blood properly. So, bad idea, blood doping. On the other end of the scale from polycythemia is anemia. Anemia means that I have some sort of deficiency in my red blood cells. That might mean I have a deficiency in the total number of red blood cells I have, or it might mean that I have an okay number of red blood cells, but that the hemoglobin present in those red blood cells is not working properly. There are different types of anemia. We're going to go over 3 general classes. The first type of anemia is if I don't have enough red blood cells total. That can happen for a number of different reasons. Maybe I just had a knife or gunshot injury and I'm bleeding out. I'm losing blood that way. That would be hemorrhagic anemia. Maybe it's because I have some sort of condition occurring that's actually destroying red blood cells that are present in my blood. That's called hemolytic anemia. Lytic means to split apart. Hemo refers to red blood cells, so we're literally splitting apart the red blood cells and losing them. Sometimes this happens from parasitic infections or bacterial infections. Sometimes it happens if people get blood transfusion and it's the wrong type of blood that can start destroying their cells. This can also happen if people undergo kidney failure because remember, kidneys manufacture erythropoietin, that hormone that's necessary for the manufacture of new red blood cells. If the kidneys begin to fail, they stop producing erythropoietin, and the overall number of red blood cells being produced drops then. You can have what is called hypoplastic anemia and aplastic anemia. These mean that I either have a decline in the total number of red blood cells being made, or I have a complete halt in the production of red blood cells if it's aplastic anemia. Sometimes these happen with exposure to certain chemicals that might destroy bone marrow cells. For example, chemotherapy can induce both hypoplastic and aplastic anemia. A second class of anemias are when I still have the total number of erythrocytes I have is still fine, but that the hemoglobin inside those red blood cells is deficient in some respect. This might happen because I'm not getting enough iron in my diet. That would be a nutritional anemia. Or it could be because I don't have enough vitamin B12 that I'm absorbing in my food. Vitamin B12 is a necessary vitamin for the manufacture of properly functioning hemoglobin. And if you don't get enough vitamin B12 in your diet, you can develop a type of anemia called pernicious anemia. So a lot of times old people or the elderly will develop pernicious anemia because vitamin B12 is a really giant molecule, and you have to have something called intrinsic factor, which is manufactured in the stomach. You have to have an intrinsic factor in order to absorb vitamin B12. And as people age, their stomachs make less and less intrinsic factor, which means their ability to absorb vitamin B12 diminishes, and they can develop pernicious anemia over time. The way to correct it is you start giving elderly people vitamin B12 injections to overcome that problem. Third major class of anemias are anemias in which the hemoglobin that's being made is abnormal. I make plenty of hemoglobin, but it's not normal hemoglobin. Maybe it's because I have some sort of a genetic condition where I don't make a proper hemoglobin chain, such as people with sickle cell disease or sickle cell anemia. They fall into this category. Then instead of making normal hemoglobin, they make a form of hemoglobin where we've just swapped out in the alpha globin, we've swapped out the glutamic acid that's in the 6th position for a valine amino acid. And just that swap of glutamic acid with valine causes the hemoglobin that's being made to not function properly. And when oxygen levels drop, the hemoglobin inside those red blood cells starts to link together to form kind of a fibrous-like protein, and it changes the shape of the red blood cell to this sort of sickle-shaped cell you see here. And they become highly inflexible, they lose their flexibility, and as a result, they get trapped in places like capillaries, and that can cause hypoxemia or lack of oxygen delivery to tissues, and it can be very painful for the person. It can, in people who have severe sickle cell disease, it can even be fatal if it's not treated. Sickle cell disease is most common among those of African American descent. And if you want to know why that is from an evolutionary perspective, ask me and I'll tell you the tale of how malaria fits into the picture. But I won't tell it here. Another type of anemia is something called thalassemia. Thalassemia occurs when the person, for some reason or other, either cannot make proper alpha chains or they cannot make proper beta chains. If they can't make proper alpha chains, they call it alpha thalassemia. If they cannot make proper beta chains, they call it beta thalassemia. Now remember what I told you about fetal hemoglobin, that fetal hemoglobin is made of alpha and gamma chains? Well, it turns out if somebody has beta thalassemia where they cannot make proper beta chains, we can actually give those people a medication that induces them to start making fetal hemoglobin instead. And this is a solution because now I'm making hemoglobin that includes alpha chains and gamma chains, and then it doesn't matter that I can't make beta. Unfortunately, if you're a person who has alpha thalassemia, there isn't such a solution as there is for those with beta thalassemia. People with thalassemia often are of Mediterranean descent. Those seem to be the individuals that are more likely to develop this kind of anemia. It is, again, a genetic disorder.